Good afternoon and welcome to the AUC Data Science Seminar Series, DSQ.
Welcome to the Atlanta University Data Science Initiatives seminar series. Today we are in for a treat to talk about cancer and data science. But before we jump in, I'd like to introduce Dr. Harris Hooker of Morehouse School of Medicine, who would like to share a few words first. Dr. Harris Hooker?
Good afternoon. Yes, good afternoon. Thank you, Dr. Washington. And I will be brief, but I honored to have this opportunity. First, to welcome our speaker for the afternoon, Dr. Dean, and to share with him and others in the Atlanta University Center just a snapshot of what's happening in the realm of cancer at research at Morehouse School of Medicine. We have a Cancer Health Equity Institute, and we know that it's centered around community and population health, but there's a large component that has to do with genomic research. And we know that genomic research has actually ushered in and it's profound new insights in this area. And it is a big part of our emerging area of precision medicine. And so it's really these therapeutic capabilities that are fundamentally really changing medicine. So as one of the students talked about moving from the bench or lab to using data science and the research, it's the same thing in the area of medicine. We have to move now from in our clinical research arena to that has to do with genomics and data science in the field of medicine as well. We also, along with that Cancer Health Equity Institute, we also have a total cancer care prevention program. And we have just in the past year enrolled, genotyped and phenotyped over 1,500 cancer patients and cancer survivors. And so there's lots of data to handle. And so we are so glad to have our Data Science Institute. I know I've changed that initiative to institute, but I'm forward thinking in that, Dr. Washington, that we're moving this initiative onto an institute. So thank you for the opportunity to share. Looking forward to hearing from Dr. Dean and forward movement with our data science initiative. Thank you.
Thank you so much, Dr. Harris Hooker. Thank you for joining us today. So again, my name is Dr. Talitha Washington. I'm the director of the Atlanta University Center Data Science Initiative, and we work across 4 institutions: Morehouse School of Medicine, Spelman College, Morehouse College, and Clark Atlanta University, as well as the AUC Robert Woodruff Library. So we are super delighted today to have Dr. Dennis Dean talk to us about how data fights cancer in the Black community. Many of us have been impacted by cancer, either by a loved one or maybe even personally. So today's talk will give us insights on how people are developing these data analysis frameworks to really uncover how to beat this really difficult topic of cancer. Dr. Dennis Dean is a principal investigator at Seven Bridges, where he leads several national scale genomics analysis projects and mentors a scientific team in the Boston area. He leads Seven Bridges collaboration outreach with the U.S. Food and Drug Administration, the U.S. Department of Veteran Affairs, Million Veteran Program, and the Patient-Derived Xenograft Network funded by the National Cancer Institute. Dr. Dean is passionate about training the next generation of scientists. He serves on the board of directors of the Biomedical Science Career Program, where he focuses on developing postdoctoral training programs and supporting STEM trainees. He is trained as a research fellow in medicine at Harvard Medical School and Brigham and Women's Hospital in the Program for Sleep Epidemiology and Program for Sleep and Cardiovascular Medicine. He earned his PhD in biomedical engineering and biotechnology and master's in computer science from the University of Massachusetts. He earned his bachelor's of science in computer science from SUNY Empire State College. And I must add, he is a fantastic salsa dancer and loves all things yoga. So with that, Dr. Dennis Dean, please grab the screen, and we are delighted to have you join us today.
Hey, let me just share my screen first. And let's get this to, so can everyone see my screen? So first of all, thank you for the warm welcome. I'm really excited to be here for a couple of reasons. I love data. It's hard to believe that I would come to that point, but there's power in data. And so what I hope to do in my talk is talk about where is the power in data and what can we do with it. And I'm going to start by just give you a little personal background perspective of how I think about data. I'm just going to give you a little background into the kind of sphere that I work in. And I want to give you a case study on preclinical trials and multi-agent cancer treatments. And to do that, I want to talk about how we develop data that can inform on future cancer treatments. And then near the end of the talk, I really want to pivot and then, take a wider set of issues. What do we have to do as a Black community of Black and Brown folks who really want to take ownership and shepherd our own data? I'll have some brief conclusions, but the goal really is to set up maybe a discussion and questions at the end. So a little perspective. So like Dr. Washington mentioned, my training is all in analytical methods. I did training in statistics, machine learning, and over time, I started gravitating towards biology and problems that could help people. I was always interested in translational research. And I think the kind of one way to kind of get a sense of who I am is kind of the problems that excite me. So I started my research in circadian biology. If we look on the left-hand side, we understand that circadian rhythms, and particularly light input, could affect behavior, and particularly how people perform. Similar to cancer, there's large data sets, which we see in the center, that have circadian rhythms and sleep. And I was fortunate to work in a lab where they have these great mathematical models for predicting human performance. There was some NASA research targeted on how we use these models to predict astronaut schedules. And one of the things that happened was I'm sitting in a meeting, giving a talk at NASA, and I'm getting grilled. I'm like, why am I getting grilled? And I spoke to a couple of people afterwards, and I learned that the person grilling me or just asking tough questions was the spouse of someone we lost in the Challenger disaster. And that really sparked an interest on how we use technology to help people. And so one of the problems in my PhD thesis was to design schedules automatically for NASA. I took a process that was manually done in a week or two and got it down to a few seconds. And the part of this that I want to share with this community is the ability to do that was really built around the fundamentals of computer science and mathematics. And so on the left side, I developed some classical representations for the problem. And then on the right, I developed a new method that allowed to solve a very translational problem that anyone could use. And this really sparked me and sparked my interest in training. And I wanted to shift a little. So in my postdoc, what I did was thinking about the association between sleep metrics, and this is just one of the problems, and hypertension or blood pressure. And so we basically, on the left side, started with 2,690 metrics, and we wanted to say which metrics were representative or predictive of blood pressure or hypertension. And so we developed methods to reduce the scope of the number of metrics. And I just wanted to do a quick aside. One of the challenges in this analysis is that people are treated with their medications. And so when you do a mathematical model, you have to adjust for that. And so we did some measures for adjusting for that. I did an automated model selection. I was really excited about this, but then one piece of the results really changed the way I think about the world, and that was the effect size. So basically, how big the effect would be for a particular population. And for African Americans and Hispanics, it was so great. It was much, much larger. And it really changed the way I think about the world and really pushed me towards precision medicine. Like, this is the area I wanted to spend my career in. So before I can go into the background of the case study, I do want to just give you a little background so we all have the general framework for where I think about cancer and data. So several years ago, then Vice President Biden announced the cancer moonshot, and the idea was to compress 10 years of research into 5 years. And the part that I really think about is, how do we generate the data sets for the analyses? And then how do we make them available so that everyone can use them? I just want to define precision medicine. You could think about taking an aspirin, it works for everyone. And then maybe you can stratify or make subgroups. But ideally, what you want to do is take the person's history, their genomics, and then say, this is the best treatment for them. There are lots of examples in the literature that this is possible. I really wanted to focus with examples with great pictures, and I just really like this slide. So this is an example in Latinos. If you look at 4 characteristics, and 3 of them are genomic related, you can see on the right side, the important part here is that these are survival curves. So based solely on some genomic characteristics, we can predict the response to chemotherapy. So this is really a good indicator that we want to be able to do more of this. I want to just define cancer very briefly without a lot of detail. So the idea of multi-agent treatments, I mentioned that right from the start. Before treatment, you have a tumor that's on the left side. You do a treatment, and then hopefully what happens is that the cancer tumor shrinks or gets smaller. But we know that over time, that cancer is a disease of change and mutation, that over time, it's possible for cancer to get resistant to the particular treatment. And that's what we're showing on the right, is that we have a tumor that's developing drug resistance. The point that I want to make here is that's why we need combinatorial treatments. We need multiple treatments, and we have to have different options so we can adapt the treatment as the tumor growth adapts. The sheer size of genomic data really necessitates doing a lot of work on the cloud. I think at Seven Bridges, we manage somewhere around 57 petabytes of data. And that's something that most people can't handle locally, or even most institutions. And so the NCI funded several cloud pilots, one of which we helped develop, which is the Cancer Genomics Cloud. And the idea about that cloud is that we could then host different types of data, genomics data, proteomics data, and imaging data. And then we can then have a whole bunch of tools that allow us to look at the data, process the data, and even collaborate across institutions. Because it's on the web, then many people, no matter where you're localized, can actually log on and look at and work with the data. And so a lot of what I'm going to talk about is how we build on top of this existing NCI-funded infrastructure. So, my case study. So I've had the fortune of working with this consortium, the NCI-funded PDXNet, and it's a consortium of about 30 PIs, 130 plus researchers. And I want to tell you a little bit about the data story. So just an overview, PDXNet is a cancer moonshot project, and the goal is to collaborate um, on large-scale development of preclinical testing and treatment of new therapeutic agents. So the idea is that we will work with new agents, identify the best ones, and then characterize them. So test them on lots and lots of models, and then generate a really large data set. And the idea is that the data set will not only inform on this particular study, but by aggregating all the data that we'll have a better understanding of the landscape of mutations or changes in the genome that might be predictive of cancer. So there's a really cool thing I gotta explain. So what is a PDX? So a PDX is a patient-derived xenograph, and you can see the example kind of in the schematic on the right. So we have a human and they have a tumor. We can sample the tumor and then implant it in a mouse, and the tumor will grow in the mouse. We could then also take a piece of the tumor growing in the mouse and then plant it in another mouse. And those are the passages 0, 1, 2 through N. And the idea here is that we can treat the mice with the same agents that we use in humans, and then we can take a look and go, are these treatments working? So this, for those of you who may have background in this area, so the idea now is this may be a little better than cell lines. And there are many reports that in order to do the human clinical trials, it's very often the request is, do you also have PDXNet data? So this is an important way to get preliminary data before we go to human trials. So the consortium, I should have mentioned, is made of a consortium of Jackson Laboratory and Seven Bridges, which leads the coordinating center, and then the 6 research groups, which I mentioned is about 30 PIs or so. And so what we do at the coordinating center is really do collaborative research that helps understand the power of PDXs. And so this is a paper just published this last February that shows that CNVs, or which are basically multiple repeats in the genome, are highly correlate with the original tumor CNV. So what does that mean? So what that means is that when we take the original CNV or the tumor in the patient and we look at the tumor in the mouse, it's actually really consistent. And so what's important about this study is the size. We looked at 1,451 PDX samples over 509 models. And so what this is saying is that, yes, this measure or the genomics in this model is very stable and could be used to study cancer. I wanted to lead, so one of the things about leading the consortium, we get access to lots of results, and they're really great treatments and biomarkers being identified. But I will leave those to the researchers to dig in as they publish them. But I wanted to just give you a preview of what you might be able to do with a large data set. And so this is a paper under review, and it's the characterization of the RNA data. So we'll just say it's related to the DNA data or to the genome for now. But if you group them all together, we actually found, and this is research led by Li Ding in Washington University, that there were 4 different classes and of subtypes. And so why is that important? So that opens up the discussion of, can we then use treatments across cancer? Maybe we can develop treatments for subgroups, and we might be able to then test them with these PDX models or other models to see how effective they are across cancers. And once again, this is the power potentially of large data. So what did we collect? So over the last 2 or 3 years, we collected 55 terabytes of data. The important part is that I think is important to know for this group is we developed new data, which was the data generated by our researchers, but we also co-localized that with data already collected by the NCI. And so this allows researchers to not only use new data, but then existing data to do analyses. And I would be remiss if I didn't say a real important part of that is really helping to develop standards. And another goal of the consortium is to get a pan-cancer view. So not just one cancer, but multiple cancers. And I think, so on the top it says 33 different cancer types. So we have multiple cancer types, but we're really enriched for breast cancers prevalent among the sample. And the reason why that's important, it starts to tell us which groupings or which parts of the data we could then explore more deeply. In addition to the genomics data, we're also collecting H&E images, which are basically histology and tumor volume data. And increasingly, our researchers are using all of these data in combination to better understand the dynamics of cancer. Now, I wanted to lead into why is this important, right? Like, so how do we go about doing this? So the first thing we do when we get these large data sets is we have to process them with workflows. So a major outcome of this group was to help develop standard PDX processing workflows. We developed about 12 of them. They're publicly available. You can see those on the left. And then the image in the upper right are the workflows themselves, visualized as a wiring diagram. And this is made possible because we use a standard called Common Workflow Language. And this standard allows us to exchange and share workflows, but also distribute the computation on the cloud. You can imagine these computations are quite large, but the CWL makes it so that we can process these automatically and then just return the results. It's important that these workflows are really documented. So it's not just enough to create them. It's really important to document the details because there can be as many as, I think some of them have 30, 40, 50 parameters that are actively used. And so really understanding the details and sharing them is important. Lastly, we want to really accommodate different types of users. And so on the right, we're showing a command line interface for executing the workflows. And we also have a GUI or a graphical interface. So let me tell you a little bit about the model collection framework. So this is the part that I spent a lot of time thinking about. When you have so many researchers thinking about sharing their data and making it useful, we really have to have a great flow. So this top part is just basically the flow and how we start with the tissue and get that to the centralized repository. And then we have to sequence it and basically generate the data for the analysis. The second part is once we get the sequencing data, we have to process it. And so in this section over here, we're generating research and research data, and that data mostly whole exome and RNA-seq. But like I said, we also have tumor volume data, H&A images, and we're also starting proteomics data. And this data has the potential for giving us even greater insights into cancer. There's a long process for processing, I should say a complex, not so much long, for data processing, metadata processing, to the point where we create data libraries. We also then have to publish them and make them publicly available in a secure way, and that's part of the dbGaP process. So the outcome of the consortium, or one of the outcomes, is then the workflows and scripts, the portals, and then what we're working towards is long-term access, and then making an interface with the NCI National Infrastructure, the Cancer Data Research Commons. There are a few things that I think were novel in how we did this, but I only want to talk about two for this talk. One is how we actually first collect the data and the metadata, or the information required in addition to the data. And this is what really makes the data useful. It has the phenotypes and the characteristics of the case, like what type of cancer, when was it taken, and that kind of information. And the second part is we really want to make the data available as quickly as we can. And so I want to talk a little bit about the PDXNet portal that we developed. So this is the PDMR model submission form. The PDMR is an NCI group that basically collects tissues in part from PDXs. And it's a form that you would kind of expect from a clinical sample, right? Information about the patient, the disease category. There are a few pages, but the point that I want to make about this is that this is the information that we can start teasing out relationships. So we may start with the genomics information, but what really makes it powerful is saying, what are the associations between the genomic information and the phenotype information? And we found developing this tool increased the quality of our metadata and what we believe the value of the PDXNet data set. And tools aren't enough, right? Like, so yes, we created the tool, but if you look at the numbers 1, 2, and 3, the tool is just one. We then have 2 additional steps where we're looking at the, where we have humans reviewing the submitted data and information. Just going through quickly about our PDXNet portal. When we look at our portal, this is publicly available. This is just the landing page. We have a quick splash. I just want to give you a sense of the pieces and the components. The first thing is we have data resources. And so these are just examples of the data resource. If you look on the left side, this is the sequencing data from the PDMR. You can see we have almost 10,000 samples. And then from these samples, one of the great things about working with the NCI is this data is really well characterized. So on the bottom, what you're seeing is the amount of data by disease type. So this allows researchers then to pull the data either to do preliminary studies or confirmatory studies, right? Like you find this result, is it verified in another test set? On the right are the H&E repository. So these are basically histology images. And we're very, very excited about this project. It just started up recently. And we already have almost 700 images. And we're getting ready to start some imaging machine learning projects. Now, I just wanted to give one analysis resource example. So basically, we have a workflow of 12 pipelines. And part of that processing is we automatically derive computational metrics. And so this is just an example of one of the metrics, and this is HRD. And for the life of me, I can never say it well, homologous recombination deficiency, but I say HRD. But we have other metrics. And so we're making these data available, in the vein of epidemiology, where we make as much information available as we can, and then we go back and say, how do we use it? So I'm just going to go here. Now, one of the things I'm really most excited about is I mentioned we have a new pilot study, Ancestry, where we want to look and characterize the ancestry component of this national data set. And so what I'm showing right now is the percentage European, but I'm going to focus on the diversity. The PDMR is 7.1 diverse. If we look at the PDXNet data, it's almost 40% diverse. And then if we add both data sets together, it's closer to 20% on the overall. So the expectation is as the PDXNet community pulls in more data and makes it available, we will be better suited and more able to study ancestry more closely and say, for African Americans, the range of mutations that we see may or may not be different, but we know that it can be different. So we want to just understand that better. So just to kind of summarize, this is just one consortium. What are the things we're doing? And so the first thing that's really important to note is, when the NCI designed this consortium, understanding that we need a diverse sample was like front and center. So the Center to Reduce Cancer Health Disparities funded 2 new research center. I just mentioned we just started an ancestry pilot study. All the researchers are really aiming to increase the data diversity. But we have one group in particular that wants to focus on prostate cancer. And there are different challenges in developing PDX models for prostate cancer. So part of the goal of this consortium is to develop these methodologies and then to share them widely. And then to just conclude, I'm really excited that we're able now to track diversity explicitly. And so what my team is working on right now is that for all the data sets and all the queries, you will automatically get the diversity component. And I want diversity to just be front and center when we start thinking about researchers or research. So I want to transition, and I ended that section with I, because I believe it's really important for all of us to own our data and own our ability to solve problems that are important for us. So when I think of cancer and hypertension, I do think about the whole world, but I also think about my father-in-law, or I think about my uncle and my aunt, both who died of throat cancer. I want to make sure that our communities are being reached. So what I'd like to do very quickly, I can't go in a lot of depth, is maybe zoom out very wide and then come back slowly on what are the things that we need to do or could be doing as a community. And it should be no surprise to people on this call that the many things that we should be doing are already being done at the Morehouse School of Medicine. So first thing, the most important thing that we need to be doing is generating numbers of diverse genomic samples. Earlier on, Dr. Harris Hooker said that Morehouse School of Medicine is actively creating new samples given the population of mostly African Americans. And this is great because what the research shows is that the types of mutations or changes in cancer differ by race. That was shown in a recent AACR health disparity report on the left. On the right, one of my favorite figures is the growth of samples. So we've gone from 1.7 million genomic samples, so that's 96% ancestry. And then in 2016, it's 35 million. But if you look really closely, and that's the figure on the right, the number of African-American samples are really small. I think it's just a couple of percentage points. So for us to really develop these new cancer treatments, we have to start here in generating the data set. We need multidisciplinary research hubs. I loved my training at the Brigham. It was just great to have so many researchers. And if you look on the Morehouse School of Medicine website, they say one of their goals in the Cancer Health Equity Institute is to get an NCI designated cancer center. And that will be so great for the community because that will allow you to not only work on the medical components, but working with all the members of the AUC will allow a reach environment to study cancer more closely. Informatics is just essential. Everything has to be programmed, even from the smallest looking at 1 or 2 samples. So really investing in informatics is important. Morehouse School of Medicine has a new Master's of Science in Health Informatics, or relatively new. This is an area that we have to look very closely about because the power of the data that is already there. So not just the new data. And so, we often say, can we ask questions? And increasingly the answer is yes. One of my favorite case studies is from Mount Sinai, and they have a chief data officer role. And that's very cool. That seems like a fun job. But what they found is that they can do clinical case studies just with the data within their medical health records. Another example, I had the privilege of visiting Optima Labs a few years ago, And they're doing the same type of thing. Let's look at this data and let's get better health treatments for our populations. You know, many of us are privileged to work in research. It's only a small percentage of the population. Cancer and health is important to all our communities. And I really think we have to work on how we reach people where they're at. I put a barbershop because that's the first place I go when I move, right? Like, where's my barber? Where's my church? Where's my minister? These are the places that traditionally our communities center around. And I believe the discussions of cancer and health have to also live there as a, not as an addendum, but just part of what we do every day. And I just want to mention very quickly, the Center of Excellence on Health Disparities at Morehouse School of Medicine is working on this, and they have a bunch of community activities that I'm sure people on the call can speak more about. Lastly, I just really want to talk about creating a full court press. There are many places we can work with. I think professional societies are a place. And I just want to mention everything on this slide came across my email in the last 2 weeks. So I didn't really have to hunt for them. But what I believe we need to do at a community is systematically understand who's doing what. So for example, there's a new initiative, let's say there's a new initiative in data science, maybe the AOC can then help coordinate new submissions and actively engage our community to participate in these. I do want to say there's some really great data sets, the multi-ethnic study of atherosclerosis, the Jackson Heart studies. In addition to cancer, really understand how we can leverage these great health data sets will be important. And lastly, I'd be remiss if I didn't say they're a really fun and interesting data science problem. What are the standards? How do we analyze? How do we analyze data across multiple institutions? Like, all of these have to be solved so that we can better understand our health. I didn't mention algorithms. Algorithms and methods are really key to what I do. And I just wanted to introduce one algorithm that is growing in use. So Seven Bridges developed a new graph algorithm for collecting information about genomics. And I'll just quickly say that the standard reference, so how you identify mutations very quickly, is basically a linear reference. But it's made primarily of a not diverse population. So the whole idea of graph is that you would collect all of this information, and then it's always available for analysis. And then in a quick study that I did at the Million Veterans Program, actually my team member did, we showed that using the graph genome seed that we're developing actually improved mapping accuracies for African Americans, and it improved variant calling in some data sets. And so what that basically means is that there's the potential for developing tools that allow us to use ancestry information better. So to just conclude, you know, there will always be new methods like PDX. They're constantly being developed. What we need to think about is how we take data collected on a small scale, putting it together so we make larger data sets. As far as owning and shepherding our data, I won't go through the long list. I will say it's really about being systematic and strategic. And lastly, we just need everyone to be involved. Cancer likely affects everyone at some point in their lives. If not you, someone in your family. So with that, I just want to thank Dr. Washington for inviting me, and I am open to questions.
Thank you, Dr. Dean, for that thoughtful presentation. So at this point, I'm going to hand it over to Bettina Gardner, our Administrative Director, who will go over our poll results. Bettina?
Hi, good afternoon. So thank you all for being here, and I'll just go through, we always ask these questions to get a better idea of who's in our audience. And so today we have mostly graduated folks and working professionals, 79% who are here. Of the students who are in the audience, they are seniors, master's students, and PhD. So those upper level students or students that are studying for their second degree. And then we have a higher percentage of females in the audience today at 68% versus 32 for males. And today we actually have more folks from the state of Georgia who are tuned in. So hello and welcome to everyone, the 38% who are from outside Georgia, but elsewhere in the US. So welcome, welcome. We're glad that you could join us from other states. And then with the questions dealing with our topic today of cancer and cardiovascular disease in the Black community, it says, what is your experience with cardiovascular disease in your life? And that can include high blood pressure, chest pain, heart attack, and or stroke. 38% said my immediate family and my extended family are affected. 9% said my immediate family only. 32% or 11 people said my extended family is affected, but not my immediate family. 4 people said my network is affected. And then 3 folks said that I know of no one who is affected. Has cancer affected your immediate or extended family? We have the 76% that says yes, 9% saying no, and 15% saying I don't know. And then what is the prevalence of screening methods in your immediate and extended family? So all of those, all recommended for screening, participate in the screening that they should. That's 24% or 8 people. Some of those recommended for screening, participate in the screening that they should. That's 12 people said that. And then 14 or 41% said that they don't know. And then our last question is, are you involved in a medical or public health profession and or studies? And so we have 56% are not in the medical field, but wanted to learn more about this. So we appreciate you coming. And then of course we have folks who are experts and knowledgeable in the field from medical and public health. So welcome to you all. Thanks for being here. And now we'll open the floor for Q&A.
Thank you so much, Bettina. So yeah, I mean, given our poll, so many of us have been impacted by cancer. My dad, Walter Rongen, has had lung cancer for over a decade and actually wrote a book about it. It's called Letters from the Land of Cancer, where he gathered letters that he wrote to family and friends as he went through and is still going through fighting with cancer. So thank you, Dennis, to share with us how data science can help us address this important topic. So I'm going to jump to the questions in the chat. So to what extent does the PDSNet dataset be available publicly to other researchers? And what is the timeline for data availability?
Yes, so my team is actively working on that. So one of the fastest ways is to just collaborate with a PDXNet researcher. But the goal is to make it all publicly available. We have the part of the process is to make it available through dbGaP. 2 groups have completed the process, and the 2 are really close. So all the groups are in process of finalizing their dbGaP applications. So data will start becoming available in the next few weeks, so not all of it, but we're expecting hundreds to thousands of samples to be available in the next couple of months. But basically, as soon as it's available, we will roll it out. In addition, once the dbGaP applications are completed, there'll just be automatic updates periodically every quarter or half year. And feel free to ping me. I can give you more details, because I can also tell you which data are going to be available and when.
Thank you. And somebody said, thank you so much. This is so fascinating. I think that attendee had to leave early. No problem. You all can find us on YouTube and subscribe to our channel. So this will be posted on YouTube as well as our other seminars. So somebody asks, with the Ancestry pilot study, are environmental factors such as soil contamination due to arsenic, which is associated with coal burning as a necessary form of heating households in the previous generations, dating back to the 1960s, and the presence of lead as well as factors associated with the data, such as diet based on available healthy food options. So just give it a little context. I believe that Professor Chef Phelps here is getting at back home in my hometown of Evansville, Indiana, there was a lot of soil contamination and arsenics in the soil, and looking at how that those environmental contaminants can really impact cancer. What are your thoughts about this?
So I think that that's a great question. And let's be clear, the progression of disease is different by location, and we know by ancestry, right? So if you're African American, you may have a whole different set of exposures. One of the reasons why I mentioned the MESA study and the Jackson Hart study is those studies are more likely to have a wider range of variables. There are definitely studies that are collecting environmental factors. This is really understood that it's important. And so it's definitely a challenge in the preclinical sense, right? Because the thought is, let's just get a quick result. And I think your question is saying, maybe we can be a little more thoughtful on the data that we collect at the start. And I think I mentioned, I really focus at the start of the data because really getting the metadata right tells us what questions we want to ask. So to answer your question, we're not collecting that now, but it doesn't mean it couldn't be collected in the future.
Someone asked, they said, I'd like to hear your thoughts on what variables or metadata in current cancer data sets are missing, in particular from people of color, right? African Americans, Latinos, et cetera. So these missing data sets, how do you think they could better inform treatment and prevention options for people of color? And how do we ensure that we have a more robust data set that can inform us on how to treat and deal with cancer?
Love this question. So when I postdoced at a lab in HMS, we were very close to the Harvard School of Public Health. And so two of the postdocs at the time, one is Chandra, forgive me for not having last names, but there was another postdoc who's now faculty at Emory. And what they have shown is that social factors are predictive of sleep. And you're going to say, okay, that's not surprising. But think about it. Like, these social factors really change our experience as people of color, that our bodies are in constant stress, potentially, over a longer period of time. Does that have effect? I would think so. But we could study that, right? We could then think through those questions and then couple them, right? And that's why I'm really excited about the Morehouse School of Medicine wanting to be an NCI established cancer institute, because then the resources start to become available to collect different types of studies and different types of data. So to answer your question, I really believe we have to look at the socioeconomic effects in relation to people of color and cancer, but also people of color are more likely to have other chronic health illnesses, hypertension, obesity, diabetes. There is going to be a complication due to that. So maybe the treatment paths need to be different. Maybe we learn there are these novel associations that just don't happen. I think it's a hard question. I think there's lots more we can collect, but I think it's really about coordinated research activities is the way to get at it.
Great. Thank you so much. I'm just going to jump ahead in the questions, if you bear with me, Dennis. I'm going to jump over to Dr. Nathan Bowen. He's a professor at Clark Atlanta University who also works in the Cancer Center at Clark Atlanta. He says, how will you increase African American prostate cancer samples in PDX-NET? It appears that there's only one sample currently showing upon query. Did we miss it?
No, I love it. Thank you. We have one group. So basically, there was 4 groups that started at the same time, and then 2 new groups that were funded by the group to increase disparities. Those 2 new groups are lagging behind the other 4 by a year or 2 years. I would say, I believe there's 35 or 50 new models from the Baylor College of Medicine group, which specifically wants to focus on prostate cancer. There's also a challenge in my understanding is that growing prostate cancer as PDXs has new challenges that haven't been completely worked out. But the main challenge is it just takes so much longer to build. So not missed, increased challenges. And it's one of the things I love about my job, right? I can call someone out and go, and it's not calling them out. It's like, what do you need to speed it up? Do we need to start more? Do we need another group? So that's why I like making the numbers available. And I think the portal's available because then there's people like you who can hold me accountable and hold our group accountable. So thank you for pointing that out. But we're on it.
Or perhaps Dr. Bowen, who works, like I said, with Clark Atlanta Center for Cancer Research and Therapeutic Development, could reach out and maybe there's a potential collaboration.
Would be happy to. And I want to tell you one of the things I would love to do with the portal moving forward. I am agile in my thinking, get it up and then see what people say. But in the next step is I really want us to present information on variants for particular genes by ethnicity. So for the population, these are the 1,000 most mutated genes. Or we also know that certain genes are mutated in African Americans more. That information I'd like to see front and center. And I can tell you my team is like, oh, here he comes. Because I'm like, can you get this up in like 2 days? And they're like, but I gotta tell you, I have a great team. Like they make everything possible. Ancestry started like really getting results October, November. I'm like, can we get it posted by December? And they gave me the results by December. We had it posted by January. So part of it is, I think as a community, is communicating the sense of urgency. As a researcher, I'm trained to like be 100% sure. We can't wait till we're 100% sure. We need to give the missing information and have the whole community add to it. So I'll leave it there. I believe it's really important to get as much information out as possible.
Wonderful, wonderful. So my friend Ann from St. Louis, Harristow, she's interested in your background in terms of what you studied as an undergraduate and what type of graduate program you did. How did you end up where you are here? And we also had, Dennis, we talked previously about how the impacts of the Black Lives Matter movement has really made a mark on how you're thinking about your research and where you want to take it. So can you share a little bit about that?
Absolutely, yeah. So if you look really closely, like there's a clock over there. It's like you put it together and there are mechanisms. I always loved how things work. I always wanted to be a scientist. So I found myself almost randomly through dancing, getting this job in the circadian rhythm and sleep group. And I loved it. And I realized that the geeky me who loves math and computers could make a contribution to research. So my undergraduate degree is in computer science, my master's in computer science with an emphasis on machine learning. My PhD is in biomedical engineering and biotechnology, but mostly biomedical engineering. And then my postdoc is basically in epidemiology. All that to say is, I was trained, so Emory Brown was my PhD advisor. He's a well-known African-American Black statistician. Or one of the things he told me is you learn what you need to solve the problem. I love problems and I love helping people. So if you give me a problem where I can help people, I'm just going to learn what I need to do. And so when I moved from epidemiology to genomics, I just read. And just to be clear, these are like papers on my list to read. I print out papers and I learn and I speak. But basically, I just like solving problems. Was that helpful? Did I answer the question?
You did. Now, what about how has the Black Lives Matter movement impacted your work on a personal level?
So on a personal level, so I have to take you through I have to take you through the process, right? So over, I would say from March to September of last year, I would have to say that this just personally, I knew as an African-American male that my PhD wasn't going to save me, my education wasn't going to save me, that for some people there is a target on my back, and that really resonated with me. But when I came out of kind of a little funk, and I would even say a little depression over this, I came back charged because what I realized is I've already been trained to solve these problems. The only thing that is required of me is to go after it every day and communicate this sense of urgency. So how's it changed? Ancestry is front and center in everything I do now. We're doing cancer, we're doing analysis. I ended with an algorithm, right? So what are the algorithms we need? It doesn't matter where you're at, what you do. It's something that I think we all have to work together to advance. So that's the answer. I can go on, but it really affected me deeply. But the only outcome is that it has me charged and reinvigorated in my current path.
Awesome. And somebody said that your comment about learning what you need to solve problems and that you love to solve problems should be broadcast to every undergraduate, to every high school student, and to all of this. Love that. And so I'll go with the final question here. What advice do you have for students who have, let's say, maybe an average quantitative skills who are interested in data science?
Yeah, so a couple of things. So I grew up in the South Bronx, really poor. The only doctor that I knew was Dr. J, right? So a basketball player. So it was, I would say there were challenges, but what I know now is that there's so many groups and communities that are there to help. So the first thing I want to say to any student is you never have to approach a path on your own. Meet people, talk to people, you'll get the right people and find a mentor, right? Because when I work with students, which I love, and students reach out to me, like I'm reviewing a PhD application this week. If I can help, I will, or I'll point you to someone. The issue isn't where you're at. And implied in that statement is that I have less quantitative skills, maybe I'm not as good. We need to get rid of all that thinking. It doesn't matter where you start, it's where do you want to go. And I think in this world of data, it's important to develop those quantitative skills. But just as important as developing the quantitative skills is the interpretation, application, or leading groups. So it's about you figuring out what do you have best to offer and develop that. I'm not minimizing the quantitative skill, but if you want to develop your quantitative skills, there are plenty of classes to do that. I'm sure the AUC can help you. But just take math, take computer science, and you take it to the level that interests you, right? Like some people don't want to be a programmer, but you should be comfortable with these quantitative and computational concepts so that you can work with a diversely trained group of people.
Awesome. So somebody asked where we can learn more about your research, and I put a link to your LinkedIn so they can connect with you and see all the other things that you are doing. But thank you, Dr. Dennis Dean, for joining us today to talk about this difficult subject of cancer and how you are employing data science to really bring valuable solutions, and also making sure that our data sets are represented. Because if we're not represented in the data sets, it's not clear that the treatments that are being developed will really benefit us, to be frank. So we really need researchers like you. We need more researchers like you to make sure that we have these diverse data sets that really benefit all of us. So again, on behalf of the Atlanta University Center Data Science Initiative, thank you, Dr. Dean, for joining us today. And hopefully this will be a start of some more discussions around cancer and data science.
Thank you so much for the invitation, Dr. Washington, and do reach out to me. I do my best to reach out to everyone quickly, and I'm particularly a fan of students. So if you have a question, just reach out to me.
Awesome. Thanks again.
Thank you.